This is why everyone is wrong about D-lactate. Hi, I'm Dr. Chris Masterjohn. I have a PhD in nutritional sciences, and I am here to help you using my biochemical and scientific expertise and my out-of-the-box thinking, harness the power of nutrients on your path to vibrant health, maximal performance, and longevity. So let's talk about D-lactate. Everyone thinks that D-lactate is exclusively of microbial origin. This could not be further from the truth, because not only is D-lactate produced by human enzymes, but I'm going to argue over the course of this video that D-lactate under normal circumstances comes in the human body almost exclusively from human enzymes and not from microbial fermentation in the gut. Now, I'm going to go beyond debunking the idea that D-lactic acid just comes from microbes during this video, and I'm going to go beyond simply demonstrating that it comes from human enzymes. I'm going to go much further than that, in fact. I'm going to show you that, number one, microbial generation of D-lactate is negligible under most conditions. Under most conditions, D-lactate comes almost exclusively from the Methylglyoxal endogenous pathway. Number two, I'm going to coin the term the D-lactate shuttle. I maintain that the D-lactate shuttle should be shown in future biochemistry textbooks as a mainstream shuttle of electrons from the cytosol to the mitochondria alongside the malate-aspartate shuttle and the glycerol phosphate shuttle. I'm going to show you how the cell will use its competing priorities, such as increasing glycolysis, conserving NAD, bypassing complex 1 when it's overwhelmed, and generating maximal ATP, how it will use how important each one of those priorities is to decide which shuttle it's going to emphasize. Even though they're not mutually exclusive, the cell can use a little bit of each shuttle together, but how much it uses of each one is going to be determined by those priorities. I'm also going to maintain that D-lactate is so important to gluconeogenesis that it could account for somewhere between 11 and 14% of gluconeogenesis under maximal conditions. I am going to maintain that the flux through D-lactate is comparable to the flux through L-lactate quantitatively, despite widespread claims that flux through D-lactate is quote-unquote minuscule, and despite the fact that the plasma concentrations in humans under normal circumstances of D-lactate are so small as to be considered infinitesimal. I'm going to show you why that is deeply misleading when you're trying to understand how much carbohydrate fluxes through the D-lactate pathway. I'm also going to maintain that oxalate is a very powerful inhibitor of D-lactate clearance, and the connection between oxalate and autism should be looked at with respect to the influence on D-lactate. I'm going to maintain that this is very important when thinking about diabetes, Parkinson's, and other neurological disorders. And if you look at the link in the description, you're going to find the written version of this video with all the live links to the scientific references. You'll also find links to some resources I have, such as Testing Nutritional Status, the Ultimate Cheat Sheet, the article that I have on optimizing CoQ10 status, and my guide on how to detox manganese, that are all very relevant to managing the status of the nutrients that are most related to the D-lactate pathway. And those include zinc, riboflavin, manganese, CoQ10, and glutathione. And to some extent, you could expand on that when thinking about oxalate to think about things like vitamin B6, vitamin C, collagen, polyunsaturated fat, and possibly, according to some hypotheses that I've laid out separately, biotin, folate, and B12. This idea that D-lactate can be made by bacteria but not humans can be found being repeated in the medical literature for decades. despite it being well known and demonstrated more than a half century ago that D-lactate is produced by human enzymes. In fact, D-lactate is normally produced from methylglyoxal. Methylglyoxal is derived from glucose in glycolysis, from acetone in ketone metabolism, or from the amino acid threonine during gluconeogenic conditions. And that methylglyoxal is then converted to D-lactate in a two-step process. It's metabolized by the zinc-dependent enzyme Glyoxalase-I to SD-lactoyl glutathione with the help of glutathione itself. And then SD-lactoyl glutathione is metabolized by Glyoxalase-II to produce D-lactate. That D-lactate can then be metabolized in the mitochondrion in an enzyme of the inner mitochondrial membrane facing the mitochondrial matrix, the inside of the mitochondrion. This enzyme is D-lactate dehydrogenase. It's dependent on riboflavin and manganese, and it converts that D-lactate to L-pyruvate. That L-pyruvate can then enter energy metabolism, be burned for energy, or can enter gluconeogenesis to make new glucose. Now, this persistent idea that D-lactate only comes from microbes was popularized in a very low-quality study from 2018 entitled Brain Fogginess, Gas and Bloating, a link between SIBO, probiotics, and metabolic acidosis. These authors took 30 people who had SIBO and brain fog, and an additional small number of people who had SIBO but did not report having brain fog. And then they measured D-lactate in their urine. They found that it was mildly elevated. We're talking, in most cases, just barely near the high normal point. In some cases, slightly above the high normal point. In no cases sufficient to actually be called acidosis. So they called it D-lactic acidosis. These people did not have acidosis based on the D-lactic acid concentrations in their urine. But in any case, they did have high normal to slightly elevated D-lactate in their urine. They then, with no control group that didn't get a treatment, they administered an antibiotic and they had them cut their probiotics. They didn't use these two separate things separately. They didn't separate the two treatments. And then they found that some of them reported that their brain fog improved, but they never remeasured the D-lactate. So they never showed that the D-lactate in the urine went down compared to the beginning of the study. So they had never had any evidence, number one, that the D-lactate was of microbial origin, and number two, that the treatment did anything to it, or number three, that the effect of the treatment on the D-lactate, which they didn't measure, because they didn't measure that, they couldn't show that it correlated with improvement in the brain fog. So they claimed that these patients were taking probiotics. The probiotics caused D-lactic acidosis, and the D-lactic acid caused brain fog, and the antibiotics and taking out the probiotics got rid of it. But they didn't have evidence of any of that because they didn't even have a control group to show that the treatment lowered the brain fog versus doing nothing. Nevertheless, this was distributed widely in popular news sites like Psychology Today and Newsweek, where they spread the new findings that taking probiotics can give you lactic acidosis and brain fog. Understanding D-lactate requires understanding methylglyoxal, and we have to go back to the origins of the discovery of glycolysis in order to properly situate methylglyoxal and D-lactate. So you can see pictured on the screen the German biochemist Carl Neuberg, and you can also see two English biochemists, Henry Drysdale-Dakin and Harold Ward Dudley. And these three people, the German guy, Neuberg, and the other two English biochemists who were working together, separately published papers in 1913 demonstrating the existence of methylglyoxal and the glyoxalase system that metabolizes methylglyoxal to lactate. Now, Neuberg devoted his career to arguing that this methylglyoxal glyoxalase system was part of mainstream glycolysis. In fact, he had written, quote, "The formation of methylglyoxal is the essence of glycolysis." Now, along comes Emden, who, alongside Newberg, was essentially a warring faction arguing for a different set of enzymes as the primary glycolytic pathway. And in 1932, Emden had put together a partial picture of glycolysis that he published as writing, "As one can see, this picture of glycolysis omits the methylglyoxal that was accepted by Newberg as the intermediary product in yeast fermentation, as well as in glycolytic lactate formation." Now, this is very important because you can see here that in formulating what is now known as the Emden-Meyerhof pathway of glycolysis, that is the standard pathway of glycolysis that you would find in a textbook, Emden was acting in dialectic with Newberg, who maintained that methylglyoxal was the essence of glycolysis. So Emden is formulating what we now know as standard glycolysis as an answer to Newberg to say, no, methylglyoxal and glyoxalase are not part of mainstream glycolysis. Mainstream glycolysis looks like this. And so when you think about that, you have to realize that methylglyoxal and glyoxalase did not deserve to be relegated into almost complete ignorance on the part of most people studying biochemistry when they were so important historically to the very discovery of glycolysis. Now, several things in history happened. Otto Warburg played a role in one of the steps of glycolysis. Then Otto Meyerhof came along as Emden passed away in 1932. Meyerhof kind of took the torch and replicated a lot of Emden's findings and consolidated them to essentially consolidate what we now know as the textbook Emden-Meyerhof pathway of glycolysis. That put methylglyoxal on the back burner, but there were other very intelligent people who were trying to say, okay, if methylglyoxal isn't the essence of glycolysis, what is it? Why is it there? So Szent-Györgyi, who won the Nobel Prize in Physiology or Medicine for his discovery of vitamin C and the role of fumarate in the Krebs cycle, who also discovered the role of actin and myosin in the molecular basis of muscle contraction. He, in 1963, published a theory that methylglyoxal's primary role was to regulate cell growth, such that methylglyoxal would retard growth and glyoxalase would clear it and therefore promote growth. And he thought that this would have utility in trying to understand how to prevent or treat cancer. That fell by the wayside largely because so many other growth factors were discovered that an emphasis on this as the primary regulator of cell growth didn't make sense. But I actually think that this is true because glutathione is needed for cell growth and replication, and methylglyoxal sequesters glutathione. So it does make total sense that the cell can control with the glyoxalase enzymes how much glutathione is sequestered by methylglyoxal, and that can be one of the regulators of cell growth and proliferation. P.J. Thornalley started working on methylglyoxal in 1984, and in his mind, he saw his role as kind of carrying on the torch of Szent-Györgyi by trying to understand the physiological importance of methylglyoxal in the glyoxalase system. He had written to Szent-Györgyi in 1984 asking him for guidance as he embarked on his research career, and Szent-Györgyi wrote back to him, "I thank you for your kind lines from May 17th and the reprints that reached me only now. It was very long ago that I worked on ketoaldehydes, Since then, I forgot most of what I knew, so to my regret, I am unable to advise you. Yours truly, Albert Szent-Györgyi. And although this letter was useless, Thornelli was very proud of it for obvious reasons and framed it, put it up in his office, and then spent the rest of his career from 1984 to the present working on methylglyoxal, arguing that methylglyoxal is, number one, the overwhelming main source of advanced glycation end products or AGEs in humans. Number two, that AGEs play a causal role in diabetes and diabetic complications. And number three, that the glyoxalase system's primary physiological role is to get rid of methylglyoxal, detoxify it, prevent it from forming AGEs, and protect against diabetes and its complications. Now, I believe this is true, but I also believe that most things that have pathological roles also have physiological roles. Now, this shouldn't be too radical. Think about how glucose is very elevated in diabetes, but glucose plays an important physiological role in the blood. Blood glucose isn't bad, just blood glucose out of control is bad. Ketones are high in diabetes. Free fatty acids are high in diabetes. Ketones play a physiological role, even in an overnight fast, even in a person on a carbohydrate-inclusive diet. Free fatty acids play an important role under the same conditions. So most things have important physiological roles, and it's only when they're outside of their natural physiological parameters that they start contributing to pathology. And I think methylglyoxal is the same thing. So in 2012, I published my doctoral dissertation, Regulation of Methylglyoxal Accumulation by Glutathione and Dietary Antioxidants. My primary research was not about the role of methylglyoxal in coordinating and serving as a substrate in intermediary metabolism. My primary research was on dietary strategies that can control its accumulation by impacting glutathione concentrations, because glutathione is important in its detoxification. However, in my dissertation, I made a unique synthesis of prior research done by other people to make an argument that methylglyoxal played an important physiological role in, number one, regulating priorities about redox status of the cytosol in glycolysis, and then number two, in acting as a gluconeogenic substrate. This belief of mine is in radical distinction to what's found in the latest 10th edition of Biochemistry by Berg, Stryer, Gatto, and Tomosko, published in 2023. To be fair, this sentence has been copied and pasted throughout multiple editions. It goes well back. It's not something that came up for the 2023 edition, but the 2023 edition still states that the production of methylglyoxal is a, quote, physiologically useless reaction. So, having studied methylglyoxal in depth, I have a very different perspective. Now, if you look on the screen, this diagram from my dissertation shows the basics of how methylglyoxal is derived and how it's detoxified and how it can play a role in gluconeogenesis by conversion of pyruvate. So glucose becomes phosphorylated in glycolysis and becomes a hexose phosphate, which means a phosphorylated form of a 6-carbon sugar, and that is then split by the enzyme fructose 1,6-bisphosphate aldolase, or just aldolase for short, into 2 triose phosphates. And on the diagram, they're just called triose phosphates, but if you know the glycolysis pathway, we're talking about glyceraldehyde-3 phosphate and dihydroxyacetone phosphate, which are interconverted by the enzyme triose phosphate isomerase. The next enzyme in the glycolytic pathway is GAPDH, or glyceraldehyde-3 phosphate dehydrogenase, which uses NAD+ to clear triose phosphates down to pyruvate. In that process, NAD+ is converted to NADH. Now, this is important because NAD+ is needed for this reaction to go down, and if the NAD+ is not there, or if there's anything else interfering with GAPDH activity, for example, GAPDH is stimulated by insulin, and there are also multiple ways that oxidative stress can lead to modifications of GAPDH that decrease its activity. or if the NAD is not there. And there could be numerous reasons for the NAD not being there. You could have a niacin deficiency. You could have a genetic defect in the ability to turn niacin into NAD. You could have a deficiency in a nutrient that's needed to turn niacin into NAD. You could have NAD being hydrolyzed irreversibly by PARP enzymes, which are involved in labeling DNA that has been oxidatively damaged for DNA repair. And since DNA gets more damaged with age, and it gets more damaged the more sick you are, NAD levels decline with age, and they decline faster in sicker people, and they need to be replaced nutritionally. And if they're not, the NAD is not going to be there. So if anything compromises and then also, if the NADH is not being oxidized effectively at complex 1 in the mitochondrial respiratory chain to restore the level of NAD+, meaning the ratio of NADH to NAD+ is rising in the cytosol, which some people would consider reductive stress. I don't like the term reductive stress because every redox reaction has reduction and oxidation. That's why it's called a redox reaction. And in general, I think people have reductive stress and oxidative stress going on at the same time. And so we should just call it redox stress. But a high NADH to NAD plus ratio would be an example of one of the redox couples in a particular compartment being excessively reduced. And that would compromise the GAPDH reaction. So oxidative stress damaging GAPDH, low insulin signaling, lowering GAPDH activity, a high ratio of NADH to NAD+, or an absolute deficit of NAD, meaning NAD or NADH is not enough of either of those in the cytosol. Any of those things are going to make the triose phosphates accumulate. The triose phosphates are what spontaneously dephosphorylate to form methylglyoxal, which in this diagram is signified as MgO, and you are always going to get some triose phosphates spilling into methylglyoxal, but anything that lowers the GAPDH reaction is going to make more triose phosphates form methylglyoxal. Methylglyoxal can also be derived from acetone, but this is going to be under different conditions. So generally when you're doing glycolysis, you're not doing ketogenesis. Under conditions of fasting or carbohydrate restriction, you're going to do ketogenesis and gluconeogenesis. Under conditions of carbohydrate supply, you're going to do glycolysis. Now, it's not binary and mutually exclusive throughout the whole body or even within a cell, but the point is, to the extent you're doing glycolysis, you're doing less ketogenesis. And to the extent you're doing ketogenesis, you're doing less glycolysis. Whereas that's not true with ketogenesis and gluconeogenesis. Those two things tend to go hand in hand. So alternatively, like to glycolysis, under other conditions, more ketogenic conditions, you could derive methylglyoxal from acetone through a two-step enzymatic pathway dependent on CYP2E1. And CYP2E1 is suppressed by insulin and stabilized by acetone, which again strengthen the association of this part of the pathway with gluconeogenic and ketogenic conditions, that is, conditions of carbohydrate restriction or fasting. And if you derive methylglyoxal from acetone, although not shown here, you will probably also be deriving it from threonine. And the derivation of methylglyoxal from threonine, especially quantitatively in vivo, is not well understood, but there's evidence that suggests that threonine in its catabolism can either go to glycine or to methylglyoxal. And what dictates which path it takes is the level of free CoA. And because fatty acid oxidation requires more CoA than glycolysis does, then you expect CoA to be more limiting under conditions of ketogenesis and gluconeogenesis, meaning conditions of fatty acid oxidation, of fasting, and of carbohydrate restriction. So in general, you will expect carbohydrate loading to promote glycolytic methylglyoxal and fasting and carbohydrate restriction to promote deriving methylglyoxal from acetone and threonine. Regardless of where it comes from, methylglyoxal forms a spontaneous adduct with glutathione that is a hemithioacetal. And glutathione is depleted by oxidative stress, and it's synthesized in response to insulin. So notice oxidative stress and insulin are playing their hand all over the place here. Glyoxalase-1 and 2 are both zinc-dependent enzymes. Glyoxalase-1 is hurt by oxidative stress and stimulated by insulin again, and that promotes the formation of SD-lactylglutathione. Glyoxalase-2 then hydrolyzes that to glutathione and D-lactate. D-lactate is then converted to pyruvate by the enzyme D-lactate dehydrogenase, which, as I said before, is an inner mitochondrial membrane protein facing the matrix, the inside of the mitochondria, dependent on riboflavin and manganese, and as we'll see, also on CoQ10. Now, this pyruvate is formed in the matrix as opposed to pyruvate being formed in the cytosol during glycolysis, but that's L-pyruvate, and it's the same pyruvate no matter where it came from, because the fate of glycolytic L-pyruvate is to go into the mitochondrial matrix and be metabolized for energy. So whether you get that L-pyruvate from D-lactate or from glycolysis really makes no difference. And one of the things that you'll notice here is this is the one and only way to derive pyruvate from glycolysis without using any NAD. So pin that in your mind, because we're going to come back to it. This completes the goal of glycolysis, which is making L-pyruvate. Well, one of the goals of glycolysis. Not all of them. It fulfills one of the goals of glycolysis to make L-pyruvate from glucose, from the glycolytic pathway, without using any NAD. Now, this figure, which is slightly modified from the one in my doctoral dissertation, shows how methylglyoxal stands in the balance of glycolysis versus ketogenesis and gluconeogenesis. So under glycolytic conditions, the methylglyoxal is derived from glycolysis, and it has a role in negative feedback inhibition of glycolysis. Now, this in and of itself is a completely fascinating discussion with very broad-reaching implications, because if you look at most ideas about glycation, advanced glycation end products are seen as the equivalent of burning and caramelizing sugar in a pan or a baked good happening inside your body. And that's looked at as random damage that has no physiological purpose. But methylglyoxal, if you just take it and you dump it on a cell and you look at what proteins it modifies, there's about 100,000 proteins and there's only about 500 that are modified by methylglyoxal. Methylglyoxal, as Thornalley has shown, is the main cause of advanced glycation end products in humans. So we're talking about the main advanced glycation end products, or AGEs, are modifications to proteins that are extremely selective, hitting up 500 of them out of over 100,000. But that's a gross overestimate of the breadth of the modifications, because when you look at those 500 proteins that might be modified, what you see is it's mostly the glycolytic enzymes that are being consistently modified. And when you zoom in on glycolysis, it's mostly one specific enzyme, which is aldolase. And remember, aldolase is the enzyme that generates the triose phosphates that become methylglyoxal. So this is a form of negative feedback on its own production, but it's way more than that. But more importantly than this, when you dump something on a cell and you look at what happens, you're not seeing what is going to happen in vivo, because in vivo, when methylglyoxal is produced, it's not randomly scattered in the cell. It's produced in a specific location, and it has proximity to specific enzymes. So in 1978, it was shown that when aldolase produces glyceraldehyde-3 phosphate and dihydroxyacetone phosphate, the two triose phosphates, the dihydroxyacetone phosphate, as it's coming out of the enzyme, It's like you give birth to the baby and you hold it in your arms and it spits up on you. It's like this. They called it nascent. It's emerging from the enzyme, like the enzyme's giving birth. And then the methylglyoxal forms from the dihydroxyacetone phosphate and inhibits the aldolase enzyme. So their methylglyoxal is not going to be in the proximity of any other enzyme to the point where it can have that degree of sensitivity to its inhibition because of the proximity. So what the general research shows is that the modification is selective even if you have pretty high concentrations randomly distributed, and it really zones in on aldolase as being the most sensitive under those conditions. But when you have conditions where methylglyoxal levels are not elevated above normal, it's going to be aldolase exclusively that gets impaired by methylglyoxal, because the methylglyoxal has such close proximity because it's being formed as the dihydroxyacetone phosphate is leaving the enzyme. So this is a very specific, potent, and powerful inhibition of aldolase in the glycolytic pathway. And what I'm going to show you in more detail later is that this is going to play a role in conserving NAD above all. And we're also going to look at how it intersects with other goals like increasing glycolysis, bypassing complex 1, and maximizing ATP production. But it's where it really shines is going to be in NAD conservation. All right, now, under gluconeogenic and ketogenic conditions, fatty acids generate acetone, which is converted in a two-step process to acetol and then to methylglyoxal through CYP2E1, as I had said before. And under those conditions, methylglyoxal is converted to D-lactate by the glyoxalase system to pyruvate, and then that can be used for gluconeogenesis. Now, what this is showing you on the right side is that if you look at a biochemistry textbook, it's going to say that fatty acids cannot generate net gluconeogenesis because they donate 2 carbons to the citric acid cycle, shown in green, and 2 carbons are removed, shown in green. And so if you add 2 plus 2 minus 2 is 0. And so fatty acids cannot net flux you any carbons towards gluconeogenesis. But where the textbooks go wrong is they don't qualify that and say through the citric acid cycle. However, through D-lactate, it's a different story. And the reality is that through D-lactate, this is a different story. Now, we're not talking about the fact that glycerol backbone from triglycerides can enter glycolysis and therefore gluconeogenesis. We're not talking about that. All the biochemistry textbooks admit that. What they don't acknowledge is that fatty acids generate acetone, which generates methylglyoxal, which generates D-lactate, which generates pyruvate, which can be used for net gluconeogenesis. 3 carbons go into D-lactate. All 3 of them can wind up being half of a glucose molecule going into net gluconeogenesis. So you really can make glucose from fatty acids after all. And as I mentioned before, there was a radiolabeling study in 1979 that showed that acetone can account for up to 11% of gluconeogenesis under maximal conditions of extended fasting. There's no studies showing how much threonine becomes glucose in humans, but in rats, it's between 2, it's basically like 2 to 4%. And if you calculate this out into grams, this is the equivalent of acetone generating 10 grams. And you could say maybe another 2 to 3.5 grams under maximal conditions could come from threonine through this pathway. So the flux through D-lactate under ketogenic and gluconeogenic conditions can reach possibly 13.5 grams, 10 coming from acetone, possibly up to 3.5 coming from threonine. All right, so that's methylglyoxal, which is needed to understand D-lactate. But now we're going to go through a short history of D-lactate itself. In the 1920s, Emden of the Emden-Meyerhof glycolysis pathway, Emden noted that methylglyoxal could form D-lactate. One of his arguments with Newberg was that methylglyoxal generates D and L-lactate. And we're going to come back to that because this only makes sense in the light of a discovery from 2014. But Emden maintained that methylglyoxal made both L and D-lactate, and that glycolysis didn't. and therefore Newberg was wrong that methylglyoxal formation was the essence of glycolysis. In 1954 and 1973 were the seminal studies that showed that D-lactate, not L-lactate, is the exclusive product of the glyoxalase system, and that seems to contradict Emden's claim that methylglyoxal generates both D and L-lactate, but we're going to come back to that at the end of this video. In 1965, the first case of D-lactic acidosis was documented in grain-fed cattle. If you look at a review, even from 2020, they're going to tell you that the first case of D-lactic acidosis in humans was in 1979 when a human with short bowel syndrome, which is a surgical resection of the small intestine, which makes carbohydrates get fermented in the colon to a much greater degree. They will say that was the first case of D-lactic acidosis in humans, but that's not true. In 1977, an unknown genetic cause of D-lactic acidosis was attributed as the cause of mental retardation in a 4-year-old. The 4-year-old had massively elevated D-lactate. They tried to sterilize his gut, they said, with 2 rounds of antibiotics. The first one did nothing to his D-lactate. The second round increased his D-lactate. And they didn't know what the cause was, but it seemed genetic. So they said, all right, D-lactate is structurally related to threonine. Remember, we've talked about threonine quite a bit today. They didn't know about this pathway. They said D-lactate is structurally related to threonine. Let's see if threonine raises its D-lactate. They did 3 dosings. One raised his D-lactate. The other 2 didn't. They said, I don't know. They said maybe it's related to threonine. It's some kind of inborn error of metabolism. It seems genetic. It's not a matter. His stool D-lactate was low. There was no basis to think that this was microbial. The antibiotic failure falsified the idea that it was microbial. And they also noted that his uric acid was chronically elevated, and they speculated that D-lactate competes with urate for renal excretion. Now, it would be 42 years until that case was solved, and we'll come back to it very shortly. But in 1979, as the reviews correctly note, or correct to the best of my knowledge, the first case of D-lactic acidosis attributable to short bowel syndrome was noted. And between that case and 2018, there were 98 recorded cases of D-lactic acidosis in humans published in the literature. Almost all of them were due to short bowel syndrome. Now, alongside, in parallel, the discoveries of the isomeric specificity of the glyoxalase reactions, where it was clearly demonstrated in the 50s and 70s that D-lactate is produced endogenously by humans, and 1977, where it was discovered that there was a non-microbial basis for genetic D-lactic acidosis in humans, medical literature was largely siloed off of this. So what's on the screen now is a quote from an article in the late 1980s from someone who's concerned with dialysis fluid used in renal dialysis, where there's a racemic mixture of L and D-lactate in the fluid, and no one ever gets D-lactic acidosis from it. So they said they were trying to study lactate metabolism to figure out why, to see how easily the D-lactate could be cleared in humans and dogs. The point that I'm bringing up here is just this quote. So he says, commercial peritoneal dialysate solutions contain a racemic mixture of D and L-lactate. Mammals produce only the L isomer from pyruvate by the enzyme L-lactate dehydrogenase, as D-lactate dehydrogenase is found only in bacteria. This is false on multiple levels, some of which were known at the time. So it was known at the time that mammals produce D-lactate through Glyoxalase-II, which has nothing to do with lactate dehydrogenase, making the fact that lactate dehydrogenase only being found in bacteria is irrelevant. It was known that there was D-lactate dehydrogenase activity in mammalian tissues. It was not known what the specific enzyme was, so we can sort of excuse the claim that it was only found in bacteria, but not really, because even if it were true that a highly specific D-lactate dehydrogenase was only found in bacteria, and even if the D-lactate dehydrogenase activity of mammalian tissues was part of a non-specific enzyme with other activity or hadn't been isolated yet, it doesn't really change the fact that the activity is there. So it's kind of false on that level as well. But the most important point is that mammals can make D-lactate, and this was known in the 50s and 70s, and somehow in the late 80s, in the medical literature, no one has any idea, even when they're very interested in D-lactate. Okay, moving on. In 2002, a putative mitochondrial D-lactate dehydrogenase was attributed to a single gene with 2 alternative splicing products. In 2002, the same year, and 2013, an Italian team headed by Lydia Dabari published 2 papers that showed that D-lactate enters the mitochondria through any of 3 putative transporters that do not transport L-lactate, where it is oxidized by a riboflavin-dependent enzyme facing the inner matrix of the mitochondria from the inside of the inner mitochondrial membrane, where the electrons from the D-lactate are used to synthesize ATP during the consumption of oxygen, and that the resulting L-pyruvate can be used for energy and may primarily be used for gluconeogenesis. In 2019, a Dutch group showed that the individual from the 1977 case report, 42 years earlier, had a defect in the gene that was identified in 2002 as the putative mitochondrial D-lactate dehydrogenase. They found a second patient with a similar excretion of D-lactate, as well as several other D-organic acids in orders of magnitude lower concentrations that seem to be products of branched-chain amino acid catabolism, although no one up to this point had ever known that branched-chain amino acids generate D-isomers. But they really provided very strong evidence, very conclusive evidence that this was the LDHD gene coding for the long sought after mitochondrial D-lactate dehydrogenase. And what they did was they said, okay, let's take the corresponding gene in zebrafish and knock it out. The zebrafish D-lactate goes through the roof. Then they said, okay, let's take wild type human LDHD, put it in the zebrafish, the D-lactate goes back down. They said, okay, now let's take our patient's putatively defective LDHD, put it in the zebrafish, doesn't do anything to clear the D-lactate. So they showed conclusively that that gene is the LDHD gene, codes for D-lactate dehydrogenase. It's responsible for clearing D-lactate in humans. And then later in 2019, an Israeli group found a whole family with LDHD deficiency that caused gout. And they said maybe just like the 1977 paper said, they said maybe the reason they have high uric acid is because D-lactate competes for urate with excretion. So they injected animals with D-lactate, and it showed that their urate went up by about 50%. And so all these things now tie together. Now, we can go back to the original 1977 case report and say, now that we know what's going on, why did the threonine experiment generate D-lactate once and not the other 2 times? Because we know that threonine metabolism to methylglyoxal to D-lactate depends on CoA status. And if you have enough free CoA, threonine goes to glycine, not to methylglyoxal. And so we can't prove that that's why it was inconsistent, but that's what you would predict, that it would be inconsistent because the path from threonine to methylglyoxal to D-lactate is inconsistent depending on CoA status. So he might just have had better CoA status 2 of the times than the other 1 time, and it makes sense that it was inconsistent. 2023, a group takes mouse D-lactate dehydrogenase and shows the crystal structure. It is a riboflavin-dependent enzyme. It's dependent on manganese, highly specifically dependent on manganese. It has no ability to run on magnesium in substitute. It is dependent on riboflavin as FAD. It has no relation to L-lactate dehydrogenase. L-lactate dehydrogenase depends on NAD. It does not depend on FAD. D-lactate dehydrogenase depends on FAD. It does not depend on NAD. 2 totally different enzymes, not evolutionarily related. Cofactors totally different. And locations are totally different. George Brooks and others have shown that L-lactate dehydrogenase has a cytosolic location as well as a location on the intramembrane space physically connected to complex 4 of the respiratory chain, but it is in the intramembrane space. It's outside the matrix of the mitochondria. It is associated with the protein of complex 4, but not embedded in the membrane. The D-lactate dehydrogenase is embedded in the membrane. It's on the inner side of the inner membrane facing the matrix. So it's completely different location as well as completely different cofactors. In 2024, the human D-lactate dehydrogenase was characterized. They showed that it donates electrons to CoQ10 in vitro, but not to cytochrome C as has been shown for plants in Arabidopsis. And D-lactate dehydrogenase is the most sensitive enzyme to inhibition by oxalate on record. Now, if you look at that, some interesting findings from a practical perspective are that about half the mutations in LDHD interfere with riboflavin binding. So you could argue that high-dose riboflavin would help promote LDHD activity in those people. Maybe it could normalize it. So maybe it could fully rescue the phenotype in the individuals whose mutations interfere with riboflavin binding. The others do not interfere with riboflavin binding, so the riboflavin is probably not going to help them. Manganese, you don't want to be deficient in it, but too much manganese can interfere with CoQ10 synthesis, so you don't want to be excess in manganese either, and it's a rough game to play. The 2024 paper, as I'll show, they show that CoQ10 accepts the electrons in vitro. Before I saw that paper, it was so obvious that this was the case, and I'll show you why it has to be the case. It's incredibly predictable that this enzyme donates its electrons to CoQ10, and I would bet a lot of money on the fact that it will be shown to do so in vivo, like it was shown this year to do so in vitro. That means that optimizing CoQ10 status is going to be very important to optimizing the enzymatic activity. You're not going to get the same effect of high-dose CoQ10 as high-dose riboflavin in the sense that you can't just oversaturate the mitochondrial membrane with CoQ10 and have it be totally harmless in the way that you can have very high concentrations of FAD floating around solubly in the cell and just get picked up by the enzymes more effectively with very little negative consequence. And then especially since manganese interferes with CoQ10 status, manganese has to be very, very carefully balanced to be trying to optimize the activity of this enzyme. Okay, and then finally, oxalate. So first of all, the fact that oxalate is found at 3 times higher concentrations on autistics, and the fact that D-lactate is neurotoxic, whereas L-lactate is not, suggests that now that we know that mitochondrial D-lactate dehydrogenase is the most sensitive enzyme to oxalate inhibition, we should definitely look at the relation between oxalate, D-lactate, and autism. And as I'll show you as we get further on, especially at the end, there's clear relations to Parkinson's and other neurological disorders as well. Okay, let's shift gears for a minute now and ask the question, is D-lactate flux significant or is it minuscule? Because in the 2023 paper in Nature Communications, they said that the D-lactate is produced in minuscule amounts. And this is not only from this paper. This is found over and over again in the literature. Even the word minuscule is found in the Israeli paper on LDHD deficiency. Even though the Israeli paper falsifies this in a way that I'm about to show you. It falsifies the idea that that D-lactate flux is minuscule. Completely falsifies it. And I'm going to show you that soon. But even in that paper, they say that the flux is minuscule. Now, the basis for this Nature Communications paper actually has a citation. And the citations are, if you just go to the description of the video, click the link, you're going to find the written article version of this, and it's going to have all of the live links to the references in the article. So you'll be able to go to whatever part you want, just click open the reference, and it's right there. Now, they cited a paper that shows that serum D-lactate is typically 11 to 70 nanomoles per liter in humans in plasma and in serum. And in serum L-lactate is 1 to 2 millimoles per liter. I would argue that optimal serum L-lactate is more on the order of 0.5 to 0.9 at rest and up to 1.2 postprandially, and then of course higher on exercise depending on intensity rather than 1.2. But let's just take the figure of 2 millimoles per liter versus 70 nanomoles per liter. A millimole is 1,000,000 times a nanomole. So when you calculate that out, this shows that normal serum lactate has an upper range of 28,571 times the upper limit of normal serum D-lactate. Now, you're going to say, Masterjohn, didn't you just prove their point? Like 28,000 times higher, that makes D-lactate look minuscule. Yeah. Okay, now I'm going to tell you this. Look at this aerial photograph of cars going down the road. This shows you the concentration of cars in that block of road. Now I want you to tell me how many cars made it from point A to point B on the day that picture was taken. Oh, you don't know? Right. Because the concentration of something never, ever, ever tells you anything whatsoever on its own about the flux of something. Because if you don't know the speed that something is going A to B, you can't judge how many molecules or cars made it from point A to B just by knowing the concentration. So that's problem number one. Problem number two is, how do you know that serum D-lactate is as reflective of whole body D-lactate as L-lactate is? I'm going to make the case to you that it clearly is not. Okay, now let's look at it like this. Let's take the Israeli paper, which funnily enough also said that D-lactate flux is minuscule. What was the blood D-lactate concentration in the people who have LDHD deficiency? Well, it was 0.7 millimoles per liter in the Dutch paper. When that same person was 4 years old, instead of 40, it was 0.6 millimoles per liter. But the family in the Israeli paper, they ranged from 1.52 to 4.7 millimoles per liter D-lactate. Okay, we are talking more than 60,000 times higher than normal. And we're also talking 3 to 5 times higher than the normal L-lactate concentration. Okay? Now, if we're going to compare this to the clearance of L-lactate, we can't use L-lactate dehydrogenase deficiency. In fact, there are different isoforms of L-lactate dehydrogenase that have a greater propensity to either create lactate or clear it by oxidizing it, but it's not a binary difference. It's a greater or lesser propensity to do one versus the other. And so if you look at even the subunit of, or even the isoform of the form of L-lactate dehydrogenase that is prone to oxidizing lactate rather than producing it, which is, as George Brooks has shown, the one that is more likely to be localized to the intramembrane space of the mitochondrion, and as others have shown, is likely to be found in greater abundance in tissues that oxidize lactate, like the liver or the heart, versus creating it like the muscle, skeletal muscle. Even when you look at a complete loss of the isoform that's biased towards oxidation, you do not see a rise beyond high normal levels of L-lactate. And that's, I think, partly because it's not 100% only oxidation for one form, 100% only reduction for the other form. And then it's also the fact that L-lactate clearance is ultimately determined by pyruvate clearance, because in order to oxidize lactate, you need to convert it to pyruvate, and then you need to get rid of the pyruvate. And so the pyruvate dehydrogenase can be the limiting bottleneck there. And therefore, what I've shown is the blood L-lactate range for pyruvate dehydrogenase deficiency, 2.4 to 4.6. You can see that the bottom is higher than the bottom of this range, but the top is a little bit lower than this range, but it's almost identical. Now, we're dealing with incredibly small sample sizes. We're talking about like 2 people over here, 6 people over here. But the point is, this is extremely powerful evidence because accumulation is production minus clearance. And if we're looking at the accumulation in the blood, we are taking out the clearance in both of these cases, which gives us a much better perception of the production. And when you take out the clearance factor, 4.7 millimoles per liter looks an awful lot like 4.6 millimoles per liter. That looks an awful lot like the production of D-lactate is comparable to the production of L-lactate. And what you usually see with the 25,000-fold difference in the blood is about the compartmentalization of these and the clearance factor that you're not looking at. Now let's take another look from a different angle. So the contribution of acetone to gluconeogenesis during extended fasting is up to 11% of the total, which is up to 10 grams per day. As I said before, if you factor in threonine, you might be able to raise that by another 2 to 3.5 grams. So we might be looking at 12 to 13.5 grams of D-lactate per day from under maximal gluconeogenic and ketogenic conditions. If we throw in the D-lactate produced by red blood cells and the extrapolation to the rest of the body, we can take PJ Thornalley's estimation from red blood cells that 0.089% of carbohydrate is turned into D-lactate. On 150 grams of net carbs, that's 133 milligrams. On 300 grams of net carbs, that's 266 milligrams. So that's an enormous discrepancy between those two figures. How are they different? Well, one is from red blood cells. The other is from gluconeogenic tissues, which is mainly the liver and to a lesser extent the kidney and maybe to a minor extent the intestine. So what's the difference? The difference is that D-lactate production is probably largely isolated to gluconeogenic tissues and is not being produced in red blood cells and other tissues that would feed D-lactate into the blood to anywhere near that extent. So how can we make sense of this? The only way you can possibly make sense of this, think about it this way. We know that D-lactate is not cleared as effectively as L-lactate from the blood. There are several experiments in dogs, rats, and bovine liver that show that somewhere between 1.85 to 3.3 times greater clearance of L-lactate from blood into liver than D-lactate. Okay, so it's absolutely impossible that a 28,000-fold differential between L and D can appear in the blood in favor of L through the liver clearing it out of the blood when the liver clears the L form better than the D form, right? So what accounts for the fact that when we block clearance, we have similar concentrations of L and D, and when we don't block clearance, we have a 28,000-fold differential favoring L in blood? How do you make sense of that? There is only one way to make sense of that, and it's perfectly consistent with the acetone versus red blood cell data that I just showed you. And that is to believe that D-lactate is made in the liver and stays in the liver. Okay? So if D-lactate is derived in the liver from glucose, from acetone, or from threonine, then LDHD can allow it to be converted to pyruvate. That pyruvate can be burned for energy, or it can be turned into glucose. The glucose can leave the liver in gluconeogenesis, But the D-lactate never leaves the liver. Why? Because it doesn't need to. There's no purpose for it leaving the liver. It can go to pyruvate, burn for energy, back to glucose. No need to put the D-lactate anywhere. Glucose can come in from the blood and generate D-lactate and generate pyruvate and generate energy. D-lactate can be taken from acetone and threonine, go to pyruvate, go to glucose, and glucose can leave the blood. Two very useful things to do with the D-lactate, none of which involve putting D-lactate into the blood. Now, how does this differ from L-lactate? Well, L-lactate, you can imagine muscle is over here and it's generating L-lactate. George Brooks has excellent information on the different shuttles. So, for example, astrocytes feed lactate to neurons. Fast twitch muscle fibers feed L-lactate to slow twitch muscle fibers. But if the muscle is producing more lactate than can be consumed by the slow twitch fiber, that lactate could enter the blood. Red blood cells are inside the blood. They obligately generate L-lactate and spit it out into the plasma for the liver to oxidize because they don't have any mitochondria. So they can't oxidize the L-lactate. So there's an obligate production of L-lactate by red blood cells going into the blood at all times that has to be oxidized by the liver or some other tissue that's going to take it up. So, for example, slow twitch muscle fiber could take it up as well. But the point is that there are many reasons for L-lactate to go out into the blood because many tissues make L-lactate that don't have the capacity to oxidize it all. Now, there's also a reason why D-lactate can't go into the blood under normal circumstances, which is that D-lactate is neurotoxic and L-lactate is not. So you can get away with having 1 or 2 millimoles of L-lactate in your blood. You cannot get away with having 1 or 2 millimoles per liter of D-lactate in your blood and get by without any neurotoxicity. So it makes complete sense that the liver, if it's a major producer of D-lactate, would be wired to not put the D-lactate into the blood. Now, what happens in LDHD deficiency? Well, this is what happens. The D-lactate accumulates in the liver, the LDHD enzyme is defective, and so it's not metabolizing the D-lactate. The D-lactate has to leave the liver somewhere. At some point, it's got to go somewhere. It can't be turned into anything useful in the liver without LDHD, and so it goes out into the blood, and that is because it has to be excreted in the urine, and the only way to get to the urine is in the blood, and that is when you get D-lactic acidosis causing neurological problems because on its way to the urine, it had to be in the blood. The blood level rose and it reached the brain and it poisoned the brain. Okay, so all of this makes perfect sense when we simply acknowledge that what has to be happening is that gluconeogenic tissues are primarily making D-lactate for their own purposes. They're primarily using D-lactate within them. D-lactate made in the liver stays in the liver. It's like Vegas. D-lactate made in the kidney stays in the kidney, and you just don't see it in the blood. That's why you have this massive 28,000-fold differential between L and D when LDHD is present, and as soon as you take LDHD away, you see similar millimolar concentrations of D-lactate in blood that you would see if the metabolism of L-lactate was backed up, that you would see for L-lactate in blood. Okay, now, is D-lactate usually derived from the gut microbiome? Well, we could look at this in a few different ways. One would be to look for the D-lactate levels found in germ-free mice and rats. I couldn't find that. Another way would be to look at the portal vein. The portal vein is the blood supply from the gut empties out into the portal vein along with blood from several other organs, and that goes into the liver. That is separate from the general circulation, which feeds the liver through the hepatic arteries and which carries blood to all the other tissues besides the liver. So since the gut empties into the portal vein, if there is D-lactate that is disproportionately coming from the microbiome, the portal vein D-lactate should be higher than the general circulation D-lactate. And I found one study in pigs, 2 papers. I can't tell if they're the same study or a different study, but they're by the same group. And they show that the D-lactate concentration is identical in the jugular vein of pigs compared to the portal vein of pigs. And that indicates that there's no signal of a greater abundance of D-lactate in the portal vein. There's no signal of, therefore, of a greater, of a disproportionate contribution from the microbiome. Now, if we want to be generous to the microbiome hypothesis, we could say, okay, maybe the intestine does not release any D-lactate from the enterocytes or any other, the colonocytes. None of those cells release endogenous D-lactate into the portal vein, whereas all the other cells, like the brain and the muscle, etc., are releasing small amounts of D-lactate into the blood. And therefore, everything that's in the portal vein comes from the microbiome. Well, okay, so about 50% of the blood supply to the liver is from the gut. So maybe 50% of the D-lactate reaching the liver from the blood comes from the microbiome then. But that's 50% of what? As we just said before, D-lactate is made in the liver and stays in the liver. So 50% of the 28,000-fold lower amount of D-lactate found in the blood reaches from the microbiome. That's, 1/56,000th of the D-lactate might come from the microbiome under normal circumstances. It's negligible. D-lactate is not coming from the gut outside of short bowel syndrome or an equivalent abnormality in the gut. Okay, now we know that D-lactate is doubled in diabetic rats. This shows you plasma and it shows you liver. Plasma is a little bit less than doubled. Liver is a little bit more than doubled. The easiest way to talk about that is just to say D-lactate is doubled in diabetes. In humans, we know clearly this is coming from the methylglyoxal pathway because we've got 3.6 to 5.9-fold greater whole blood methylglyoxal, depending on whether you're looking at type 2 or type 1 diabetes. And we have similar proportions of methylglyoxal-derived ages that are higher in diabetes. So clearly, this is signaling that the methylglyoxal accumulation and therefore the D-lactate generation from methylglyoxal through endogenous human enzymes is driving this increase in D-lactate in diabetes. And so diabetes being one of the most common afflictions of modern society, therefore is telling you that we should be looking at the methylglyoxal pathway to explain anything common about moderate rises in D-lactate and possible cognitive problems from that. We should be looking at the endogenous methylglyoxal pathway rather than the microbiome as our first mode of explaining that. All right, now let's answer the question, what is the physiological purpose of the D-lactate pathway? So in addition to what I had noted in my 2012 doctoral dissertation, which I went over in the beginning about the importance of methylglyoxal and D-lactate in regulating the redox status of the cytosol during glycolytic conditions and serving as a gluconeogenic precursor, we can now use additional research to answer this in more detail. D-lactate enters the intramembrane space through voltage-dependent anion channels, or VDACs, which are very non-specific channels that allow many organic acids, NAD, NADH, ATP, ADP, ions, etc., all go in through VDACs. The inner membrane has very specific transporters that allow specific organic acids or specific ions to get into the matrix. And there exist several to get D-lactate into the matrix. In the matrix, the riboflavin and manganese-dependent enzyme LDHD is on the inner side of the inner membrane facing the matrix. D-lactate is in the matrix, gets converted to L-pyruvate in the matrix. Now, the transporters are all putative. They showed functionally that these appear to exist, but we don't know the exact proteins and genes behind them and so on. And there are 3 putative D-lactate transporters, none of which transport L-lactate. D-lactate has a proton symporter, meaning that D-lactate and a proton, meaning a hydrogen ion, meaning H+, meaning acidity, is brought in together with D-lactate in the same direction. And it has an antiporter with malate and what's called a 2-oxoacid antiporter, which the malate antiporter allows D-lactate to come in while malate goes out, allows D-lactate to come in, and the 2-oxoacid antiporter allows D-lactate to come in while pyruvate or oxaloacetate exit. And right now, you should think about hydrogen ions are associated with glycolysis, which is acidic. So you're going to want hydrogen ions coming in if D-lactate is coming in from glycolysis. But if D-lactate is coming under gluconeogenic conditions, you're going to want malate, pyruvate, or oxaloacetate to exit, because cytosolic malate, pyruvate, or oxaloacetate are all gluconeogenic precursors. All right, so in the DiBari papers from the Italian group in 2002 and 2013, they looked at what D-lactate was doing relative to the respiratory chain. So I'm just going to briefly review this standard diagram of the respiratory chain. You've got the intermembrane space on top. You've got the intermitochondrial membrane being shown with the complexes being embedded. You've got the mitochondrial matrix on the bottom. Complex 1 oxidizes NADH, and it pumps protons using the energy from the electrons in the NADH. The electrons get shuttled to CoQ10. They do not go through complex 2. They get shuttled through CoQ10 to complex 3. Complex 3 takes their energy, pumps more protons. Complex 3 donates the electrons to cytochrome C on the intermembrane space side of the membrane, which delivers the electrons to complex 4, which pumps more protons. At complex 4, the electrons are glued to molecular oxygen, which becomes water. And then those protons, they have backflow through complex 5, which is ATP synthase, which produces ATP from ADP and phosphate. Complex 2, as this diagram misleadingly suggests, that free-floating FADH2 drops its electrons off at complex 2. That's false. Nothing like that ever happens. FADH2 and FAD are inside this enzyme. Succinate should be shown here, being oxidized to fumarate as part of the citric acid cycle. Inside this complex 2, a bound FAD becomes a bound FADH2. No, there's never free-floating FADH2 dropping its electrons off here. Inside this complex 2, FAD becomes FADH2, taking electrons from succinate, while succinate becomes fumarate, and then the electrons are passed to CoQ10 from complex 2. They never go anywhere near complex 1 when that happens, and then they pass through the rest of the chain as just described. Now, one thing to note here is that the protons are what's used for the ATP synthesis. So you get a lot more protons pumped. To be precise, you get 6, as shown in this diagram, instead of, 10 instead of 4. 10 instead of 6. You're missing 4. And what that means is that if you drop NADH off here, you're getting 40% more ATP, not 40% more. You're getting, let me put it this way. Complex 1 is responsible for 40% of the ATP you're making, and complexes 3 and 4 are responsible for 60%. So instead of getting 100% of the ATP, when you come in through CoQ10 to go to complex 3, you're getting 60%. That's a 40% loss of ATP when you come in the middle of the chain. As we'll see, there are benefits to coming in the middle when complex 1 is overwhelmed. Specifically, if complex 1 does not have the capacity to handle the NADH that's coming in, there will be reactive oxygen species generated, and the NADH will get backed up. If the NADH gets backed up, you back it up in the cytosol, you cause problems for the GAP-DH reaction to go forward in glycolysis. Rising NADH to NAD plus ratio compromises all of the reactions that depend on NAD plus, even if they have nothing to do with energy metabolism. For example, NAD plus is responsible for DNA repair. If you don't have NAD plus, you can't do that. There are many things that depend on NAD plus. And so if complex 1 gets backed up, the rising NADH to NAD plus ratio is a problem, and then you're generating more reactive oxygen species. Reactive oxygen species, if they weren't held in check, would damage the proteins and lipids in this respiratory chain and permanently compromise energy metabolism. The defense against that is to use negative feedback in response to reactive oxygen species as a signal to shut down energy metabolism broadly. So you will shut it down broadly to avoid making more reactive oxygen species, to avoid long-term damage. Any of those things is negative, right? So there are going to be cases where if complex 1 is overwhelmed, you want to come in through complex 2, or you want to come in through something else that donates electrons to CoQ10 to bypass complex 1, but you do so at the expense of getting a 40% drop in the total ATP that can be generated. All right, now what the Dabari group showed with the electron transport chain was that when they looked at the electron flow with D-lactate, if they blocked complex 1 with rotenone, it did not do anything to the ability of D-lactate to be oxidized. They showed that oxygen was being consumed and that ATP was being generated, implying that the electrons are flowing all the way through complex 4 so oxygen can be made into water, and that's allowing hydrogen ions to synthesize ATP in complex 5. Now, if they blocked complex 3 with antimycin, they blocked the ability of D-lactate to do any of those things. So what that implies is that D-lactate dehydrogenase is dumping the electrons off somewhere between complex 1 and complex 3. Is it dumping it off at complex 2? Obviously not, because literally nothing else dumps electrons off at complex 2 except succinate. It's obviously not doing that. Further, it itself is membrane bound. So it can't be donating electrons to complex 3 because it's embedded in the membrane, and it's not embedded in the membrane as a supercomplex with complex 3. So how would the electrons get to complex 3 unless they were going through CoQ10? It is beyond obvious that this is an enzyme that directly donates its electrons to CoQ10, as shown in 2024 in vitro. But it has to be the case if you try to liken it to any known existing enzyme, or you try to integrate it into anything we know about how the respiratory chain works. So let's take this diagram from a CoQ10 review. There are 5 other known enzymes that are situated in the inner mitochondrial membrane. All of them depend on riboflavin, and all of them donate their electrons to CoQ10 in the inner mitochondrial membrane. These include sulfide detoxification through SQR, which is sulfide quinone oxidoreductase. That's involved in hydrogen sulfide metabolism. It takes electrons from hydrogen sulfide, it passes them through FAD to make FADH2 inside the enzyme that then donates the electrons to CoQ10, turning it from the oxidized ubiquinone to the reduced ubiquinol, regenerating FAD inside the enzyme, and then that dumps them off at complex 3. Proline dehydrogenase works exactly the same way. Faces the matrix, inner side of the inner membrane. Proline from the matrix becomes P5C in the matrix. FAD's inside the enzyme. FAD becomes FADH2, dumps the electrons off at CoQ10, the squiggly tadpole in the picture, regenerates FAD inside the enzyme. Ubiquinone becomes ubiquinol, dumps the electrons at complex 3. ETF dehydrogenase faces the matrix in the same way, embedded in the inner side of the inner membrane in the same way, has FAD bound inside the enzyme in the same way. It receives electrons from electron transferring flavoprotein, or ETF. ETF receives electrons from fatty acid oxidation, the oxidation of branched chain amino acids, the oxidation of lysine and tryptophan, and the scavenging of methyl groups from sarcosine and dimethylglycine, which are methylated glycine metabolites that have buffered extra methyl groups. ETF dehydrogenase is needed to rescue those methyl groups and rescue that glycine at a later time to scavenge them. Electrons from any of those sources reduce FAD to FADH2 inside the enzyme. Then FADH2 delivers them to CoQ10, turning ubiquinone to ubiquinol, regenerating FAD inside the enzyme, and ubiquinol drops them off at complex 3. These green enzymes, dihydroorotate dehydrogenase and the mitochondrial glycerol-3 phosphate dehydrogenase, which is part of the glycerol-3 phosphate shuttle, do exactly the same thing. The only differences are their substrates and products are in the intermembrane space instead of the matrix. They're on the outer side of the inner mitochondrial membrane instead of the inner side. Dihydroorotate dehydrogenase has an additional difference that instead of using FAD, it uses riboflavin in the form of FMN. But otherwise, it's exactly the same. D-lactate dehydrogenase is obviously doing the exact same thing. The D-lactate is shuttled into the matrix of the mitochondrion. D-lactate inside the matrix is the substrate. It becomes L-pyruvate, the product inside the matrix. Inside the enzyme, FAD becomes FADH2. Inside that FADH2 then drops the electrons off at the ubiquinone inside of the inner mitochondrial membrane, which becomes ubiquinol and donates them to complex 3. I'm going to argue that this constitutes the D-lactate shuttle, which should be described in biochemistry textbooks as operating alongside the malate-aspartate shuttle and the glycerol phosphate shuttle. And it's not acting in exactly the same way, but it is achieving similar priorities in similar ways. Now, what you'll see in descriptions of the malate-aspartate shuttle is that NADH electrons from glycolysis cannot enter the matrix. So they reduce oxaloacetate to malate. The malate carries those electrons into the matrix, and then it's oxidized back to oxaloacetate, producing NADH. So NADH electrons made it into the matrix through the organic acid malate that carried them there. And so the idea is that the electrons are shuttled on an organic acid. That allows glycolytic electrons from NADH to become mitochondrial electrons on NADH without NADH being able to transport through the membrane. All these other organic acids here are ways, they're just fuel to keep this system operating. The key point is that NADH electrons are carried on malate into the inside of the mitochondrion to generate mitochondrial NADH. The glycerol phosphate shuttle does a similar thing, but as we saw several slides ago, this is using the enzyme mitochondrial glycerol-3 phosphate dehydrogenase, which is in the outer side of the inner mitochondrial membrane, and it takes electrons, turns FAD to FADH2, drops them off at CoQ10, turning ubiquinone to ubiquinol, then that goes to complex 3. But if you look at this in the broader picture, there's a cytosolic or cytoplasmic glycerol-3 phosphate dehydrogenase that's taking NADH electrons from glycolysis, NADH becomes NAD+, that converts dihydroxyacetone phosphate to glycerol-3 phosphate, and then those electrons are not becoming mitochondrial NADH. They're being added to FAD to ultimately put into the ubiquinone pool to go straight to complex 3. So the malate-aspartate shuttle is generating mitochondrial NADH that is oxidized at complex 1, generates 100% of the ATP that could be generated from that NADH. The glycerol phosphate shuttle is going straight into complex 3 from the CoQ10 pool. That's losing 40% of the ATP that could have been generated by that NADH. Why would you do that? We'll get to that in a few minutes. But the point that I want to make is there are some differences between what I'm proposing in the D-lactate shuttle and the way that these two items operate. So the big difference is that D-lactate is not involving NADH at all. Both of these other shuttles are described as means of getting glycolytic electrons from NADH into the mitochondrial matrix. So they both involve the oxidation of cytosolic NADH in both of these shuttles. And that's not happening here. But think more carefully about this. What is happening here? What's happening here is that this D-lactate has the electrons that would have gone onto the cytosolic NADH had the GAPDH reaction proceeded. But as an alternative to it, the gap DH reaction was avoided, and this is carrying as an organic acid into the mitochondrial matrix the electrons that would have gone on that NADH. So some biochemical purist who's writing a biochemical textbook might say that's not really a shuttle like this is, because it's not taking electrons from glycolytic NADH. But I would say, think about the overall objective, which is to get the electrons from the glycolytic pathway into the mitochondrion. This shuttle is doing that because the D-lactate has the electrons that would have gone on to NADH. And instead of generating NADH like the malate-aspartate shuttle is doing, it's generating FADH2 inside of an intermitochondrial membrane enzyme that is putting those electrons directly into the ubiquinone pool that's putting them into complex 3. So it's fulfilling the same objective of getting those electrons into the mitochondrion as these two are, and it's doing so in the manner of the glycerol phosphate shuttle in the sense of bypassing complex 1 and going straight to the ubiquinone pool to drive those electrons into complex 3. It's doing it a little bit differently because it's not oxidizing NADH anywhere. It's not involving NADH, but that difference is not any bigger than the difference between the malate-aspartate shuttle and the glycerol phosphate shuttle. For example, the malate-aspartate shuttle involves an organic acid carrying the electrons directly into the mitochondrial matrix. The D-lactate shuttle does involve an organic acid carrying the electrons directly into the matrix, and the glycerol phosphate shuttle does not. So if you look mechanistically, there are some differences between the D-lactate shuttle and these two, but that difference, namely being that it carries the electrons that would have gone onto NADH instead of the electrons that did go onto NADH, that difference is not any bigger than the difference between the malate-aspartate shuttle and the glycerol phosphate shuttle, which is that the malate-aspartate shuttle involves shuttling an organic acid directly into the mitochondrion, and the glycerol phosphate shuttle does not. So to me, these three shuttles are very analogous in their objectives. They're very overlapping, and the differences between any two of them are not any bigger than the differences between the other two. So I believe that the D-lactate shuttle, which I'm coining this phrase in this video right now for what I believe is the first time. I want this to wind up in the next edition of Berg Biochemistry, right alongside these two shuttles. Okay, in order to situate this in the context of the other shuttles, and I'll be developing a master class with Masterjohn energy metabolism course that goes into this in much more detail. Let's take methylglyoxal out of the picture, and let's look at how you would use malate-aspartate versus glycerol phosphate shuttles in a simplified manner. So glucose has, as it goes into the glycolytic reactions, there are two ATP consuming reactions that lead to two phosphates being put on fructose 1,6-bisphosphate, which gets split into two triose phosphates by aldolase. In those reactions, acidity is generated. 2 ATP generates 2 ADP plus 2 H plus. This in this diagram, there should be a parenthesis around here. The 2 should be here. This is 2 protons that are being generated in these early glycolytic reactions. Now, that's acidic. And because it's acidic, there's dramatic feedback against these two phosphorylation reactions. If acidity builds up in the cytosol, it will counteract these two reactions, and that will mean that glucose does not get phosphorylated. If glucose does not get phosphorylated, not only does glycolysis not go forward, but the glucose can't stay in the cell. Phosphorylation of glucose is what traps it inside the cell. So if the glucose can't be phosphorylated, your blood glucose will go up in reaction to the acidity. So we do not want these reactions to go forward generating acidity without neutralizing that acidity. All right. Fructose 1,6-bisphosphate gets split by aldolase into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, the two triose phosphates. NAD plus is used to clear them down through the rest of the pathway in the GAP-DH reaction. That generates more acidity because every time you consume NAD plus to generate NADH, you generate NADH with a proton. So there's 2 NAD plus with generating 2 NADH and 2 protons. Now you've generated 4 protons. In the next reactions, you have 4 ADP and 4 protons that become 4 ATP. So you generated 4 protons up through GAP-DH that was acidic. Then you neutralized all 4 of them in the 4 phosphorylation reactions that generate ATP at the end of glycolysis. The typical textbook split is in the energy consuming phase, which is up to aldolase, or arguably aldolase plus triose phosphate isomerase, which interconverts the 2 triose phosphates. And then the energy generating phase is the production of NADH and ATP. But you could also split this at a different point, where the acidity generation phase is the first half of the glycolysis plus GAP-DH. And the alkalinizing phase is everything after the GAP-DH reaction, or specifically starting with the ATP generating steps. By the time you get to 2-pyruvate, you have made glycolysis net pH neutral because 4 protons were consumed in the ATP generating reactions to neutralize the 2 that were generating in the ATP consuming reactions at the beginning and the 2 that were generated by the GAP-DH reaction. The production of lactate is alkalinizing because it consumes 2 more NADH plus H plus to make 2 NAD plus. But and I'm taking this model from the work of George Brooks, which shows that L-lactate is almost always made from pyruvate in the cytosol. It travels into the intramembrane space, and as described before, although this diagram is not highlighting this part, the mitochondrial L-lactate dehydrogenase is physically connected to complex 4, uses the energy from the reduction of oxygen to water in complex 4 to fuel the oxidation of L-lactate back to L-pyruvate in the intramembrane space, and then the pyruvate is transferred into the mitochondrial matrix for energy metabolism. Because this reaction is reversed, it happens in the cytosol, then it's reversed in the intramembrane space, the net effect on cytosolic NAD+, NADH, and acidity is actually neutral because the intramembrane space ions and organic acids they're essentially continuous with the cytosol because the VDACs in the outer membrane of the mitochondria are very general and non-specific, and so there's essential equilibration of these things. However, in practice, what this does is it creates spatial distance between NADH, acidity, and glycolysis. So there's actually evidence that L-lactate dehydrogenase in the cytosol physically connects to GAP-DH and facilitates direct transfer of NADH from GAP-DH to lactate dehydrogenase so that the oxidation of glyceraldehyde-3 phosphate down through the glycolysis pathway is coupled to the direct reduction of pyruvate to lactate. And what that does is that immediately gets NADH away from GAP-DH, immediately feeds NAD plus directly into GAP-DH, and that allows that reaction to rapidly continue, and that's called substrate channeling. Now, the production of L-lactate in the cytosol and shuttling into the mitochondrial space, this allows the proximity of the NADH and the acidity to be moved into the intramembrane space. And as soon as the acidity and the NADH are regenerated, they immediately have access to the malate-aspartate shuttle and the glycerol phosphate shuttle, which can then move that acidity and that NADH into the mitochondria, into the matrix, and then that effectively removes it from the cytosol. So you have a back and forth in this alkalinizing NAD plus generating effect of making lactate in the cytosol. But by the time you use these shuttles, you have allowed the combined production of lactate and dropping off of electrons at these shuttles. You have allowed that to make glycolysis net alkalinizing. All right. And the reason that's important is because acidity shuts down glycolysis. NAD deficiency shuts down glycolysis. So why would you use the malate-aspartate shuttle versus the glycerol phosphate shuttle? This dotted arrows are signifying the choice that you could go one way versus the other. The reason you would do that is because if complex 1 is overwhelmed and the NADH cannot be oxidized there, then the NADH will build up. It'll build up in the endomembrane space. It'll build up in the cytosol. It'll shut down the GAPDH reaction. That also will ultimately shut down the absorption of acidity in the NADH oxidizing reactions, and therefore acidity will build up and it will shut down the phosphorylation reactions. Glycolysis shuts down, blood glucose rises, glucose utilization falls. So what you want is that up to the capacity of complex 1, you want to use the malate-aspartate shuttle. And then also in the mitochondria, if you're trying to shove NADH into complex 1, you generate reactive oxygen species. Reactive oxygen species shut down energy metabolism because if they don't, all of it, if they don't shut down energy metabolism, then they'll build up and they will damage the mitochondrial membrane and the respiratory chain proteins, and that will cause more long-term damage to energy metabolism. So when complex 1 traffic is too high, you funnel through the glycerol phosphate shuttle. This has the advantage of bypassing complex 1 to avoid NADH building up, to avoid reactive oxygen species building up. It has the downside that it generates 40% less ATP. So it's your plan B, not your plan A. But if you go in with plan A and plan A fails, you're in big trouble. So your plan B is what saves you. Now let's look at what methylglyoxal does different. So first of all, methylglyoxal has negative feedback against aldolase. It shuts down the triose phosphate production. Why is that important? Well, if you come back here, and here you can say, okay, NADH to NAD plus ratio is rising in the cytosol. That's going to shut down GAPDH. Yes, it's going to shut down GAPDH, but not totally. If aldolase keeps making the substrates for GAPDH, the substrates rise and they force the GAPDH reaction to go forward because of substrate supply, and that worsens the high NADH to NAD plus ratio. That worsens the acidity problem in some respects. And so the methylglyoxal inhibition of aldolase is conserving NAD more effectively than allowing the GAPDH than allowing the triose phosphates to accumulate. So a portion of methylglyoxal can inhibit aldolase. If it's bound to aldolase, it's not going to do anything else. But a portion of methylglyoxal can make D-lactate. Notice that making D-lactate does not involve NAD. D-lactate carries hydrogen ions into the matrix through the D-lactate proton symporter. And then as with the glycerol phosphate shuttle, the D-lactate is being oxidized to pyruvate in a way that directly inputs electrons to the ubiquinone pool that go to complex 3. Now, the big differences here are that you have a much more effective conservation of NAD because you shut down triose phosphate production and you do not drive the GAPDH reaction forward through substrate supply, and you generate D-lactate and ultimately mitochondrial pyruvate in a way that completely avoids the need for NAD. If you go back to these other shuttles, you can't get down here without NAD. And you can't use either of these shuttles without NAD. But you can use methylglyoxal to generate D-lactate that becomes pyruvate in the mitochondrial matrix with no NAD whatsoever in the cytosol. So you conserve NAD much more effectively. You get around using NAD. You spare it much more effectively. The downside is that even though you are mitigating the acidity by bringing protons into the mitochondrial matrix, glycolysis is still net acidic because you are not completing the last half of glycolysis, which is the alkalinizing phase. So if we compare these three shuttles, the malate-aspartate shuttle leads to net alkaline glycolysis. It maximizes ATP production. It cannot bypass complex 1, and it requires NAD plus and NADH. The glycerol phosphate shuttle leads to net alkaline glycolysis. It leads to 40% less ATP. It bypasses complex 1 when it's blocked, and it requires NAD plus and NADH. The D-lactate shuttle leads to net acidic glycolysis. This is a big downside. It leads to 40% less ATP. This makes it the same as glycerol phosphate shuttle. It bypasses complex 1 when it's blocked. That makes it the same as the glycerol phosphate shuttle. It does not require NAD or NADH. That is completely unique, and it adds extra conservation of cytosolic NAD by Methylglyoxal inhibiting aldolase. That's completely unique. So where D-lactate shuttle really shines is in extra conservation of NAD, in its ability to work even in an NAD/NADH deficit, and in trying to decide which shuttle to use, the cell is going to think about what its needs are. If it wants glycolysis maximized, it needs to use these top two shuttles. If it wants ATP maximized, it has to use the malate-aspartate shuttle. If it wants acidity maximally buffered in the cytosol, it's got to use these top two shuttles. But if its biggest concern is NAD conservation, then the D-lactate shuttle will have tremendous utility. In reality, the cell is not using one or the other. They're not mutually exclusive. It's using all three all the time, but it's going to take those priorities and how they balance out to decide which shuttle to put more emphasis on at any given point. In gluconeogenesis, it's a lot easier, a lot more straightforward to explain what's happening. So in acidity, where you have protons that are accumulating in the cytosol, you want to use the D-lactate proton symporter. In gluconeogenesis, you bring D-lactate into the mitochondrial matrix. You export malate, pyruvate, and oxaloacetate. These are gluconeogenic substrates. D-lactate becomes pyruvate, goes out. If you convert D-lactate to pyruvate, that becomes pyruvate that exits as new D-lactate comes in, and you wind up getting malate, pyruvate, and oxaloacetate in the cytosol, where they can all be used to synthesize glucose. All right, so let's add a nuance here. Does Glycolysis-III spare the brain? So unbeknownst to me at the time, months before I finished my dissertation in 2012, a novel human glyoxalase unrelated to Glyoxalase-1 and 2 was discovered and dubbed Glyoxalase-III. The gene for the DJ1 protein was discovered as an oncogene in mice in the 1990s, and mutations in it were associated with early onset Parkinson's in the 2000s. It's now dubbed the PARC7 gene because of this. Alongside these discoveries in the 1990s, a gene in E. coli was dubbed Glyoxalase-III for its ability to convert methylglyoxal to lactate without the use of glutathione or any other cofactor. So Korean researchers, realizing the similarity between the mammalian DJ1 gene and the E. coli Glyoxalase-III gene, they demonstrated that the mammalian DJ1 enzyme has cofactor-free glyoxalase activity, and they showed that in 2012. In 2014, they showed that it converts methylglyoxal to L-lactate rather than D-lactate. This is true for the human enzyme, but not the plant version. The plant version in Arabidopsis makes D-lactate. The authors noted that DJ1 is highly expressed in the brain, where LDHD expression is very low, and suggested that putting L-lactate rather than D-lactate in the brain is very important for neurological health. Notably, plants don't have brains, making the neurotoxicity relevant, which might be why they don't have the isomer specificity that the human enzyme has. Indeed, a tissue-based map of the human proteome showed in the next year that LDHD is most highly expressed in the liver and kidney, with considerable expression in pancreas, moderate expression in heart and liver, in heart and skeletal muscle, and low expression in the brain and many other tissues. PARC7 has high expression in most tissues, including the brain, so it's not specializing in the brain. LDHD is low in the brain, PARC7 is high in the brain, reflecting that you want a lot of L-lactate and not a lot of D-lactate in the brain. That makes sense. The gene for Glyoxalase-II, which generates D-lactate, has a somewhat similar pattern as LDHD, but the expression in the brain is not minimal, even though it's lower than in liver and kidney. So the picture doesn't perfectly fit that idea. And further, the gene for Glyoxalase-1 is fairly evenly expressed across tissues, and it even has higher expression in brain than kidney and liver. Now, we can say from this that now that we know about Glyoxalase-III, Emden must have been right when he said that methylglyoxal generates both L and D-lactate back in the early 1900s, even though Glyoxalase-1 and 2 were shown to generate D-lactate exclusively in the 1950s and 70s. And then on top of that, Now if we synthesize this, what we see is methylglyoxal has a fast and spontaneous adduct formation with glutathione. The glutathionated portion can go through the glyoxalase-1 and 2 system to become D-lactate. The free portion can go through the Glycolysis-III enzyme to become L-lactate. The cell can then prioritize what it wants. If it wants more L-lactate, or it wants more D-lactate, or if it wants to sequester methylglyoxal as SD-lactoyl glutathione, then it can change the expression of those enzymes. For example, high glyoxalase-1, low glyoxalase-2 sequesters methylglyoxal as SD-lactoyl glutathione. High expression of both glyoxalase enzymes generates more D-lactate. Low expression of glyoxalase enzymes, higher expression of Glyoxalase-III or PARC7 generates more L-lactate. Now, in the brain, I think we have to do more research to dissect this in different cellular levels, but consider these points. Astrocytes feed neurons to L-lactate as their primary fuel. D-lactate dehydrogenase skips over complex 1 to feed electrons into the CoQ10 pool. This generates 40% less ATP than feeding electrons into complex 1. Blocking complex 1 with rotenone causes Parkinson-like dysfunction in animal models. Mutations in PARC7, which block the conversion of methylglyoxal to L-lactate, cause early onset Parkinson in humans. And D-lactic acidosis, but not L-lactic acidosis, causes neurological dysfunction from the unique neurotoxicity of D-lactate. Well, how do you synthesize all this? To me, it looks kind of simple. D-lactate interferes with L-lactate in the brain by driving electrons into a 40% less efficient means of ATP production. I'm now thinking that it's not the high NADH to NAD ratio that makes blocking complex 1 with rotenone cause Parkinson-like dysfunction in animal models. Rather, it is the drop in ATP that you get when you don't use complex 1. And the brain really wants L-lactate funneling through complex 1 to generate maximal ATP in neurons. And when you drop that 40%, you get Parkinson-like dysfunction or you get other neurological problems. All right, so what you want is the versatility to use the D-lactate shuttle when complex 1 is overwhelmed and NAD needs to be conserved, but you don't want complex 1 overwhelmed in the first place, and you don't want NAD running low in the first place, and so you don't want to force neurons to bypass it. Okay, some practical considerations. This is a biochemistry lesson, so it's not primarily practical, but Quest has a plasma D-lactate test. Home lactate meters are not isomer specific. You have to do them right, so you want to wash your hand well, swab it well, get a big drop of blood, remove at least the first one, if not the first 2 or 3 drops before you measure lactate with the finger prick. Even if done properly, you have to realize that it's a mix of venous, capillary, and arterial blood, even if you remove damaged tissue, cellular debris, and so on with the procedure I just described. And so it's not going to be perfectly lined up with a venous blood draw from the forearm. But in general, if you see a big difference with a higher home lactate finger prick measurement on the meter taken around the same time as a venous draw for an L-lactate measurement through Quest or LabCorp, that's probably going to indicate a significant amount of D-lactate in the blood. You can always verify that with a Quest D-lactate test. Of course, D-lactate is expected to mostly be in liver and kidney rather than the blood, so you're not always going to see the implications with a blood test. Nutritionally, the glyoxalase enzymes depend on zinc, insulin, and glutathione. D-lactate dehydrogenase depends on riboflavin, manganese, and CoQ10. See testing nutritional status, the ultimate cheat sheet. Does CoQ10 deserve a spot on your longevity plan? And my How to Detox Manganese guide for relevant ways to manage the status of those nutrients. Since D-lactate dehydrogenase is the most sensitive enzyme to inhibition by oxalate, we can think about the contribution of oxalate to autism involving D-lactate clearance. We can think of strategies to lower oxalate like supplementing B6, avoiding vitamin C, megadosing or collagen, reducing PUFA, consuming 300 to 400 milligrams of calcium per meal, reducing dietary oxalate, and perhaps optimizing status for biotin, folate, and B12, as is indicated by my alternative hypothesis about how pyruvate carboxylase might be able to convert oxalate into carbon dioxide. All of these things become practical considerations. All right, so the bottom line, D-lactate is primarily made endogenously in most circumstances, not by the microbiome. I, in this video, hereby dub the D-lactate shuttle I want to see this in biochemistry textbooks alongside the glycerol phosphate shuttle and the malate-aspartate shuttle. The D-Lactate shuttle operates alongside these shuttles to help the cell make decisions about its priorities when it balances conserving NAD, maximizing glycolysis, reducing cytosolic acidity, or bypassing complex 1, as it decides what to do with those different priorities and how they balance out. It's going to decide which shuttle to place emphasis on. Where the D-Lactate shuttle really shines is that it can operate in an absolute deficit of niacin or NAD and NADH, and it is superior at conserving NAD, even though it is inferior at removing acidity from the cytosol. D-lactate is an important contributor to gluconeogenesis, somewhere between 11 and 14%, somewhere between 10 and 13.5 grams per day under maximal conditions could be the flux through D-lactate to new glucose. D-lactate should be taken seriously for its potential role in Parkinson's and in neurological problems generally, for its role in diabetes, and for its extremely underappreciated roles in glycolysis, gluconeogenesis, and the respiratory chain. We know that D-lactate flux appears comparable to L-lactate from comparing L-lactate dehydrogenase deficiencies to comparing D-lactate dehydrogenase deficiency to pyruvate dehydrogenase deficiency. That shows you that when you block the downstream metabolism, the blood levels are comparable instead of the 28,000-fold difference you see between them. That is because D-lactate is made in the liver and stays in the liver. It's made in the kidney probably and stays in the kidney probably. And it's not fluxing through the blood. And it's not fluxing through the blood because it's neurotoxic. And you don't want to do that unless you have to get it into the urine. Oxalate powerfully impairs D-lactate clearance, so D-lactate should be investigated as a potential link between oxalate and autism. Oxalate-lowering strategies should be seen as ways to improve D-lactate clearance and reduce its potential role in diabetes and neurological disorders. And see the sections on riboflavin, zinc, manganese, and glutathione in my Testing Nutritional Status, the Ultimate Cheat Sheet, as well as my articles on CoQ10 and detoxing manganese for managing the relevant nutrients. Look forward to more Masterclass with Masterjohn energy metabolism lessons on the D-lactate shuttle in the context of the other shuttles we discussed and the roles of L-lactate. And finally, if you want to trace back the scientific references, go to the link in the description to the written article. All the references are laid out as live links in the written article. And that's all for today. I hope you found this useful, fascinating, and interesting. And that's D-lactate. Go spread the truth about D-lactate by liking this video and sharing it. And if you're not already, don't forget to subscribe.